Requesting everyone to settle down, please. We are kickstarting session 2. Thank you. Thank you. We heard our dignitaries speak about the vision that they have for the Pharma Innovation 2047, and it was really a goosebump moment for me when Mr. Jain said that the ambition is to get 100 new products out of Indian innovation. And we don't see a reason why we can't get there. I think we just need that focus, we need that passion, and we need all of us to connect the dots really. And to get more into the specifics of that, that's where we have the next session brought to us by session curators Dr. Pawan Singh and Dr. Seema Pai. And there we are going to see really how can we unlock the future with some of the advances that we see. And I want to take a moment to introduce our distinguished curators for this session. So starting with Dr. Pawan Singh, he's a doctor by training from GS Medical College and MD DM from PGI Chandigarh. Currently working as VP and Head of Clinical Development for Kashiv Biosciences, but has previously worked with a plethora of organizations such as Biocon, Glenmark, Sun Pharma, DRL, and Pfizer, holding roles of increasing responsibilities in clinical development. He has published several peer-reviewed national and international, in several peer-reviewed national and international journals. He's also a member of IEC at Homi Baba Cancer Hospital and Research Center, Muzaffarpur, and a senior advisor to Doctors for You, a pan-India humanitarian organization with international presence. So really a multifaceted personality, and thank you so much, Dr. Pawan, for moderating this session as well as for your contributions to the steering committee. And then I want to introduce Dr. Seema Pai, who is, apart from her role as Senior Director of Clinical Site Operations for India and Southeast Asia and Pfizer, also holding a very responsible position of being the President of Indian Society for Clinical Research, which is the nodal body for clinical research professionals in India and driving innovation in every aspect of clinical research. Seema is a member of the leadership team for Asia Pac organization of Pfizer, and of course India leadership team, and board member of Pfizer Products India Limited. So she brings not only the research element but also the business element for the table. And she's of course experienced in various aspects of clinical research such as virtual monitoring, decentralized trials, innovation site and study solutions. And she's an alumnus of the Grand Medical College and TN Medical College, both very esteemed institutes in Mumbai. And apart from ISCR, also actively contributes and collaborates with various bodies such as OPPI, ICMR, BIRAC, CDSA, and several other government initiatives. So it's my pleasure to hand over to you, Seema, to take the session forward. Thank you so much.
Thank you very much, Sunila. I think there is a problem with this. Yeah. Thank you, Sunila, for the kind introduction. I think post the networking break, where I hope everybody has refreshed themselves, we are moving into our next session for the next gen therapeutics. And without much ado, I would like to highlight that this session, just like the previous session, is going to be power-packed, because we will have 3 exciting talks and with very eminent speakers in this particular session. And also we are going to look at the how of it. So far we have been hearing all the messaging around what was there in the first session as to the why of what we are doing today. This is going to more focus on the how of it, especially when it comes to the next generation of therapeutics that are going to be innovated in this country. And as we heard Sunila say, there is a very ambitious goal that has been laid out. So without much ado, I will like to introduce our first speaker for this session, Dr. Shridhar Narayanan. He is the founder, director, chairman, and the CEO of the Foundation for Neglected Diseases Research. He's a very seasoned drug discovery leader with more than 20 years of experience across discovery and development in multiple therapeutic areas. And he holds a degree in pharmaceutical sciences from the University of Mumbai and is a PhD from the Ohio University, and postdoctoral training in neuropharmacology from the UCLA. He is the founder, director, chairman, and CEO of the Foundation for Neglected Diseases Research, which is a not-for-profit organization which was established in 2014. And it has managed to advance medicines for diseases of the developing world, which we talked of in the morning, is of so much importance to our country. And he's also the co-founder of Peptris Technologies, which is an AI/ML-driven drug discovery company. And from the morning, we are hearing about AI/ML being introduced and a new search engine which is being developed. And this is where I think his insights will be very, very important for this session. He has previously served as the Vice President and Head of Innovative Sciences at AstraZeneca India, and he has led the TB and the malaria program, advancing multiple clinical candidates and executing in-licensing and out-licensing deals exceeding more than $180 million US. He's a senior technical advisor for innovation to the IPA, and as Dr. Jain said, this particular innovation arm is very, very important to take us in our journey forward as a country. And he has been a mentor to startups, to academia, and he has authored more than 50 publications and has over 25 patents to his credit. So with that, I will hand it over to you, Dr. Narayanan, for your talk, which is on the engineering future drugs, innovating in India for the world. So what do you, sir?
Very good morning to all of you. Thanks, Seema, for the kind introduction, and thanks to you and Anirban for inviting me to be part of this power-packed session. Yeah. It's been a fantastic morning listening Dr. Renu Swaroop talk about the vision and further being extended by KG, Sudarshan, Sanjeev. I think it's been a fantastic session in the morning. I hope that I can continue that interest in innovation and start talking about my interest in the crossroads of technology and pharmaceutical sciences. While the title of the session is Next Gen Therapeutics, I'm going to talk a little bit about the technology bit and how technology is going to influence the development of some of the therapies that we are developing. So while Seema introduced, she mentioned about my roles in different organizations. And though I pride that the foundation is my child, I today will talk about Peptris Technologies, which is the AI/ML company that I'm working on. So I've kind of put my title a slightly different one. So I want to talk about the crossroads of pharma and engineering and how engineers are getting into the pharma field more than pharma people themselves, right? So the title of the talk is Engineering the Future Drugs, Innovating in India for the World. So all of you know about this drug discovery process. I don't think I need to introduce that topic, but just as a rehash, right? For people who are not familiar with this, the traditional drug discovery process takes anywhere around 12 to 15 years with an investment of more than a couple of $1 billion. And Dr. Khurakewala mentioned about the time it takes for drug discovery to actually happen and the patience that is required as a drug discovery scientist, right? So my experience has largely been in the initial part, the preclinical testing part, and to some extent in the clinical development up to phase 2. And I've moved molecules from scratch all the way till phase 2 myself. So I'll talk a little bit about how we are using technology to kind of hasten this process, right? If you look at AI-based drug discovery, AI is a buzzword today. Everybody talks of AI in every aspect of life, not only in pharma, but every aspect of life, right? And I think on a regular basis, we are familiar with Gemini, ChatGPT, the Perplexity, and all of those things. So where AI plays a role in drug discovery. So it has multiple areas where it could have an impact. One is target identification, that is identifying which target would be responsible for a particular disease. The design of the drug, that is the initial hit identification stage, a lead optimization, and also into preclinical testing. So there are multiple aspects of AI which could potentially have an influence on each of these areas of drug discovery. I'm not going into development. I'm just talking about the discovery bit yet, right? So AI can obviously influence the entire continuum going from discovery to development to marketing to everything, right? But I'm going to focus on this aspect. This is what we're doing within the organization. So the way normally, if you talk to a chemist, the way their representation of a chemical molecule or a drug would be as a smiles. I'm not sure if you can see the carbons, oxygens, the chlorides, all of it, right? So if you look at a drug molecule, it's generally represented in a smiles fashion. Now this smiles fashion, when you write, it is not conducive for an algorithm to read it, right? So what we have done is we have come out with our own possible representation of the smile structure, which is patented. Now you could have other ways in representing the molecule like a 2D structure or a 3D structure or a surface one. What we have done is we have done a graphical representation of the molecule. So the algorithms that we have created identify each of these molecules as graphs. And the algorithm allots those graph-like structures to the molecules such that if you look at that right side of the screen, it's filled with dots. Those represent the chemical molecules. And if you look at the chemical space that is available, it is about 10 to the power 60, right? It's a figure that's absolutely unimaginable. And if you were to congregate those dots or sort them out using an algorithm, you would find that molecules which are similar, I'm not talking about similarity in terms of only the chemistry, but similar in terms of their biological properties, would all congregate together in certain clusters, right? And that's what the algorithm is able to do, is identify patterns based on these graphical representations, which could put molecules which are similar, quote unquote similar, right? And put them into these clusters, which can then create really novel spaces, right? So what we've done is we have taken about 1 billion molecules from the large database that is theoretically available. Today we have organizations like Enamine or Molport, which can make these molecules based on click chemistry, right? And that was the Nobel Prize that was given out, I think, a couple of years ago for chemistry, where the technology is there. You have Lego-like building blocks, and you can put together these blocks and make molecules whichever way you want, right? And what we've done is we work very closely with these organizations. We've developed our own syntax of representing these molecules. We work with these organizations which can make these molecules and come up with really novel molecules. So if you look at where AI works in the whole drug discovery space as well, all of you have heard of AlphaFold. We are DeepMind, AlphaFold. Now we've got AlphaFold 1, 2, 3, right? So we use our algorithms on top of AlphaFold, right? So is it that everything is open source? No. We source from the open AI whatever is available, and we lay our algorithms on top of that to actually create efficiencies within the system for us to look at new molecules and how we can actually make better predictions of the activity of some of those molecules. There is optimization, there is generative AI, and this is the big piece, right? So generative AI is the bit where you learn from all of the representations and you start creating molecules on your own. And this is where some of these organizations like Enamine come into picture, because we have now created Lego blocks which necessarily don't theoretically fit together. But based on our algorithm, we have found that they can actually be synthesized and they could actually work for certain therapeutic targets. So based on that, we do some work in that side. And the fourth one is the predictive modeling. So all of these pieces are part of our in-house suite of algorithms that we use to actually do some of the work in drug discovery. So if you look at AI and value addition at this point in time, where it is adding value, you have the target identification, a lot of the phenotypic screening, a lot of the omics biology, drug repurposing. is a major play where AI comes into picture. And I'll give you a case study of how we have used AI for drug repurposing and actually move a drug forward for a disease which is a rare disease indication. So where do we work? What space do we work in the drug discovery continuum? So the pointer is not seen there, but what we work is the early discovery, hit identification, the lead optimization, and the preclinical development. So that's the sweet spot. That's the sweet spot where we use our AI algorithms. The model is use the literature, programs like AlphaFold to work on the disease hypothesis and the target identification part, do the work in the early discovery to preclinical development, and then find partners to do the clinical development and commercialization bit. And it's a very standard model that we use of licensing milestones, royalties, and revenues. And depending on the therapeutic area, depending on the molecule, we find licensing partners at different stages of development. So what we do is 3 different phases, right? So we have this 10 raised to power 60 chemical molecules. We do something called as a unsupervised learning, right? And this is how ChatGPT actually learns. It takes words, random words, and the context of sentences which is used in the literature, puts them together and derives meaning. And it can now form a new sentence based on that. So what we do is we do this unsupervised learning of this large space of about 30 billion molecules. We bring them down to about 100,000 molecules. So this is a very coarse thing, right? So if anybody's used the old radio, right, you'll know, pehle wo station ghumane ke liye we'll do the coarse thing. And then you have a fine adjustment where you kind of get the frequency right and you start hearing the words properly, right? So we do that coarse filter. So we get the station right first. And then we apply models which are supervised learning models, which will enrich the data set. So from 100,000, we bring it down finally to about 100. And this is an iterative process which is happening in silico. So there is no cost involved except for the compute cost, right? All of this is theoretical at this point of time. So imagine what is happening today, right? Before AI came in, you had all the large pharma companies doing this work through something called as high throughput screening, where they would have libraries of physical compounds and they would have a target and they would work on that target. They would bombard that target in a biological assay and find new use cases for it, right? Or starting points for that particular target. What we are doing is we are reducing that million molecules to about 100. And that 100 also happens in an iterative process. So our first assay would normally be run with about 30, 40 molecules. We find hits from that, go back into this loop, find new molecules, right? And what we have done is, because of that, we have increased the chemical diversity. So if you look at the statins, all of them would probably look alike to a chemist, right? There is limited chemical space that is available. Freedom to operate is a big challenge. What we've managed to do with this is create the kind of chemical diversity that is absolutely unthinkable, right? And which is where I think it's a very, very powerful tool. So finally, we come down to about 100, which goes into the screening. And this is for novel molecules. Now for the repurposed assets. So in addition to doing the screening for the target, what we are able to do is we are able to create, if you were to imagine an Excel sheet of all the proteins that are characterized by AlphaFold versus all the approved drugs, right? So let's take a random number. AlphaFold can do about 30,000 proteins. Imagine an Excel sheet with 30,000 proteins and about 10,000 approved or drugs in clinical development, right? Can you actually make sense of that table? It's going to be impossible to do that. So what we managed to do is do some of these things in a very novel way. Obviously, I can't disclose how we do that. That's proprietary intelligence. So what we call it is an augmented intelligence, not just artificial intelligence, but augmented intelligence. And using that, we are able to predict new use cases for existing drugs, right? And these are use cases which you would never have thought of. So the first case that I want to talk to you, and I'll tell you about one scenario which we have done for a disease called Duchenne Muscular Dystrophy, right? Now, Duchenne Muscular Dystrophy is a rare disease, affects boys primarily, 1 in 3,500 cases seen, progressive loss of muscle force and autonomy, and it results in severely impaired quality of life and premature death, right? So the average lifespan is anywhere around 18 to 23 years, right? So you have the diagnosis being done around age of 3 or 5. 70% of the boys lose ambulation by the time, seems to be moving on its own. Artificial intelligence at work. So, well, there's 3 minutes on the clock, so it probably wants me to hurry out. That's okay. So you have 70% of the boys lose ambulation. By 15, they are on ventilator support, and by 20, they've lost their life, right? And while there is a huge monetary component to it, there's a huge emotional component to it as well, right? Imagine a family going through this, a 10-year, 12-year-old boy. Oh, wonderful, thank you. When you look at it, the kind of dependence that a teenager has to have on a primary caregiver, and the primary caregiver in most cases is the mother. For a teenage boy to request his mother to help him go wash himself, that kind of emotional drain is something that can have a huge, huge impact, right? And that's the quality of life bit that we are talking about. So today, what is the treatment that is available? The only treatment available is steroids in India, right? Globally, there are some exon skipping, some gene therapies that are available. They are not very successful, very expensive, right? There is a recently approved drug called Givinostat, which is on the market in the US. It costs about $700,000 per patient per year, right? So $700,000 roughly translates in today's currency to about 6.5 crores per patient per year, right? So what we are trying to do is try to develop something which will replace both of these, or at least add on to this to improve the quality of life. So the way we are working on it is we are looking at upregulating a protein called utrophin, which is something that is there from childhood in all of us, but it comes down by the age of 5. So we are trying to see if we can increase that as a surrogate protein, which will help retain some of the muscle function. We are reducing inflammation, and we are also helping the muscle to regenerate, right? So what we did is we did the process that we talked about. I'll talk about the repurposed one, right? So we had the whole list of repurpose that went through it. We predicted about 4 actives, of which 2 of them actually worked in the biological assays. So what we did was some early biochemical assays and also preclinical testing in mice, which is a model which is called D2MDX, which is the dystrophin model for this disease in mice. What we saw was a significant benefit across all the muscle-related functions, right? So the first one is we put the mice on a treadmill and have them run. So if you see the wild-type mice, which is your normal mice, they run about the maximum of about 350 meters in a matter of half an hour. And if you look at the DMD ones are able to run maybe half of it. And when we give our drug, there's a significant improvement that you start seeing. You see improvement in the grip strength, where the muscle tone is much better. You start seeing improvement in the hanging test, where you let the mouse hang from a wire, and I'll show you a video of that. There are various other parameters that we measure, pre-treadmill and post-treadmill, because once you've stressed the muscle, the strength of that muscle goes down significantly. So if your drug is able to improve the muscle strength in that, there is a possibility that it will improve the overall quality. So based on this, so this is a video of the mice actually in action, and you can tell me which one is the drug-treated one and which is not. So you have the wild-type controls on the extreme left. The center one is the disease mouse, right? And the last one is our drug-treated one. I'll let it play for about 10 seconds. So the disease mouse falls down, and you have our mice continuing to go on. And we've done this repeatedly, number of times now. And very clearly, the drug seems to improve this in mice. Mice are not men, right? And we definitely want to see whether this translation can happen. And that translation bit is what we are currently doing. We have applied for an orphan drug designation for this. That should come hopefully by the next month. We are also filing a US FDA application with our partner. So this molecule has already been licensed to a company called Therapeutics. They are moving it forward in development. We'll be doing a clinical trial application to the US FDA as well. So hopefully within the next 6 to 12 months, this molecule should be in clinical development. It's a repurposed drug, which has been on the market for almost 20 plus years now. Very safe. So we don't see a challenge taking it from the indication that it has been approved for to a Duchenne muscular dystrophy indication. And if this actually works, then it's an absolute game changer, both in terms of the improvement in the quality of life and also in terms of the affordability of the drug itself. So what we've also managed to do is do a whole lot of build a pipeline of molecules across therapeutic indication. And this is where AI is really, really impactful, right? You are not going to be restricted to one therapeutic area. You could use the same algorithms to actually identify drugs across a spectrum of diseases. So we are doing all the way from oncology, and this is one on cancer metastasis, alopecia, rare diseases, name it, and you could actually diversify into that therapeutic area. We talked about building capability, building capacity. So we work with the ecosystem across the world, right? And the beauty is we are an 8-member team. We have raised about 1.3 million so far to do all the work that we've talked about and taken a molecule all the way to clinical development, built a pipeline of molecules. We are currently in between our fundraise. I have some of my angel investors in the audience. So thank you very much for supporting the cause. We talked about funding requirements. A company which does the kind of work in the US would probably raise about 100 million. We're having a tough time raising even 7, 8 million, right? And that's the challenge. While we talk about everything that's good, and I completely acknowledge the support that is there from the government, the regulators, all of them. If you're a startup today, and we are probably the first AI startup from India who's managed to do a licensing deal, it's still a tough road ahead, right? So if there's anyone in the audience who wants to contribute to this cause and be an investor, please do meet me afterwards. Happy to take any money that is there. I want to leave you with the vision, and this is the vision we share at IPA. So today's presentation has been about multiple hats, right? Want to leave you with this message. Firmly believe that this is a deliverable goal, right? 100 new therapeutics from India by 100 years of independence, not a challenge. We already heard about a dozen of them. We are well on our way. And I believe that as a country, we can truly make that difference, not only for our own patients, but also for the world. Thank you very much for your attention.